really okay. It's been really an inspiration for my earlier work. So I've during my PhD, I was one of the inspirations of my work in enhanced biodegradation and microbial degradation of pesticides. He was one of the pioneers in the area of microbial degradation of pesticides, especially starting with atrazine, but also all other type of herbicides. And in the last, I would say, 15 years or so, 10 years ahead, you moved to antibiotics and veterinary drugs. So there, he has been the pioneer in identifying the possibility, the potential of microbes to degrade veterinary antibiotics when they are repeatedly exposed to veterinary antibiotics. And since then, he's been very active in this area, looking at the interactions of veterinary antibiotics with the soil microbiota. So I will give the floor to Ed. Ed has been working in Agri-Food Canada, but for those of you that you didn't hear the conversation we had, he has now moved to INRA in Dizon, working in the group, working with Fabrice Martin Laurent and Laurent Philippot, I suppose. And he's going to be there for at least the next 3 years. So it is a pleasure to have him with us, and he's a member of the advisory board of Aristo as well. So please, Ed, the floor is yours.
Thank you, Dimitros. Very pleased to be with you and very pleased to be affiliated in any way with this Aristo project, which is very interesting. Now that I'm in Europe, hopefully we'll be able to interact more often than we've had in the past. So I did retire from Agriculture Canada, which is the Federal Ministry of Agriculture, after 40 years. And I'm very pleased to have been successful in getting money to work here in France and work with European colleagues over the next few years on irrigation with reused water. So in many parts of the world now with climate change, water is limiting and there's an issue with food security. So there's a move afoot to increase the use of wastewater effluent in agriculture. And so what we're going to do is look at some aspects of whether that practice would increase the likelihood that there's transmission of AMR from crop production systems through the food chain back to people or not. And if there is a problem, how do we manage it? But so for today, the next few minutes, I'm going to speak about the fate and impacts of antibiotics in agricultural soil. And I'm screening through here. If it's not working for you, just let me know. And I've turned off my camera because I'm getting a message that my internet is unstable. So for the purposes of this presentation, when I talk about antibiotics, we're talking about antibacterial agents. There's often confusion between the words antibiotics and antimicrobials. Antimicrobials, more broadly speaking, to chemicals that are used to kill viruses, bacteria, fungi, parasites that are problematic for human or animal or crop health. So we're talking about antibacterial agents. And so an antibiotic is a type of substance that's active against bacteria. Of course, these medications are widely used in the treatment of prevention of bacterial infections in human medicine and in agriculture including both animals and crops, and in the case of aquaculture, fish, for example. So antibiotics may either kill or inhibit the growth of bacteria. And so these antibiotics are a wide range of chemistry. They can be natural chemicals that are produced as secondary metabolites by bacteria or fungi, for example. They can be semi-synthetic derivatives of those natural antibiotics. And there the example, the classic example would be penicillin. And we're now, I think, in the 4th generation of penicillins where modifications have been made in order to defeat resistance to penicillins. And some antibiotics are completely synthetic, such as the fluoroquinolone antibiotics. So typically these chemicals were designed to be effective in a person or an animal, for example. And so the pharmacokinetics, the absorption, distribution, metabolism, and excretion has to be such that the chemical, that the antibiotic is within the host long enough to be effective, and it has to be distributed in the bloodstream of the host or at the site of infection. So typically they're not particularly lipophilic, for example, and they have to be non-toxic to the host, which will limit the potential non-target effects that one might be concerned about when these chemicals get in the environment. These chemicals have a wide range of physical properties, stability, their characteristics with respect to absorption to organic matter, for example, whether they're charged under typical environment conditions. And so, as with other pharmaceuticals and pesticides, you can't think of these as being one common thing. You need to consider their environmental fate and impacts on a case-by-case basis, a chemical basis, or a class basis. And just say that in soils, in the environment more broadly, metabolism or co-metabolism is arguably likely the major driver of environmental dissipation. And so, don't think of these things as being POPs, for example. In terms of the broadly mechanisms of action, again, we're looking for targets here that are specific to bacteria and would not be found in eukaryotes, mammals, for example. So a lot of these antibiotics act on ribosomes, the machinery that is used for protein synthesis. So for example, the macrolide, tetracycline classes of antibiotics. Other antibiotics inhibit cell wall biosynthesis and integrity. Penicillins would be an example there with respect to peptidoglycan biosynthesis and breakdown. Some chemicals act on DNA replication, DNA gyrase, for example, the fluoroquinolone antibiotics. And finally, others can act on the biosynthesis of essential intermediate molecules. An example there would be the sulfonamide antibiotics that act on an enzyme involved in the biosynthesis of tetrahydrofolate. And so again, what I'm trying to emphasize here is when you think of these bacterial specific targets, they are not designed to be non-toxic to a host, and that would narrow the potential non-target effects on soil or environmental biology. So you would not think that these chemicals are going to be problematic for bears or penguins, possibly plants that have chloroplasts that are vestigial gram-negative cells, but still you would design your program to look at impacts with this information in mind. So in terms of the key pathways of antibiotic entry into agricultural soil, and thinking about this in contrast to pesticides, for example, certainly a key pathway is the use of human or animal or poultry waste as soil amendments, as fertilizer. And we'll talk about that in a minute. So that would be a major route of entry into soil. Another route of entry would be the use of antibiotics as pesticides in crop production. And it's not the case in Europe, to my knowledge, but certainly in many other jurisdictions, in North America, for example, and certainly in many lower income countries, that antibiotics are widely used to prevent or treat bacterial infections of crops. So for example, in Canada, kasugamycin and streptomycin would be used to treat fire blight in the production of apples, for example. In other jurisdictions, oxytetracycline, streptomycin could be used to treat bacterial pests of tomatoes, for example. So there's this whole use of antibiotics as pesticides. And finally, a third example, which is more of an emerging one, would be irrigation of crop production systems with wastewater effluent. So sewage treatment effluent that has the potential to contain antibiotic residues as well as biological contaminants. But these would be the three routes of entry that you need to think about. And so the first one has to do with the use of fecal fertilizers in agriculture. And if you look at this bottom of this image, you see three pictures. The middle one is a crop production system growing maize in this instance. And if you think about what we do fundamentally in agriculture, we apply nitrogen, phosphorus, potassium to soil. We plant a seed in that soil. We ask the crop to photosynthetically fix carbon from the atmosphere, making biomass. Then we harvest that biomass from that field and we feed it directly to people who consume that, or we feed that biomass to animals that we then consume in the form of meat or eggs or milk, for example. And a portion of those nutrients are excreted by people and excreted by animals. And so in a circular economy, for a number of reasons, it's very desirable to recycle that fecal material back on the ground. It recycles and conserves nutrients, improves soil structure and water holding capacity, which is very important in a time of climate change. And it's just generally a good idea. But of course, these fecal materials can contain biological and chemical contaminants that we need to think about when we're evaluating potential risk and how we can manage that risk. And what you see in this table here is a rank ordered on the basis of concentration list of pharmaceuticals that were detected and quantified in a biosolid sample, so a sewage sludge sample, from the sewage treatment plant in London, Ontario, where I used to work. And this was an extract prepared, I think, in 2011 or 2012. So at the top left-hand corner of the table, triclocarban, which is a biocide, which in this biosolid sample is at a concentration of roughly 6 milligrams per kilogram. And you see a whole list of chemicals going down this list that are decreasing in concentration. Those chemicals that are highlighted in yellow are antimicrobial or antibiotic or biocidal. So you have a number of fluoroquinolone antibiotics, ciprofloxacin, again, the top left here, norfloxacin, ofloxacin, all at milligram per kilogram concentrations. The minimum inhibitory concentration that a physician would be looking to get in your bloodstream of ciprofloxacin is of the order of 3 milligrams per kilogram. Here we're at 6 milligrams per kilogram of these biosolids. And you see other classes of chemistry, tetracycline, macrolide antibiotics. And so when we saw this, we became concerned that putting these material onto agricultural land at these concentrations is high enough that it may be having an impact on soil microbiology. So that's one route of entry, these fecal materials. The other route of entry, one of three, is use of antibiotics as pesticides. We have very little knowledge of how much of what antibiotic is used in crop production, but there was a paper published a couple of years ago by Phil Taylor and Robert Reeder, where they interviewed agronomists that are advising farmers in low and middle income countries on how to handle pests in their crops. And the conclusion from this survey was that far more antibiotics are being used than we think maybe. So this is certainly a reason to look at this in more carefully. And finally, the third route of entry is effluents from sewage treatment plants being used to irrigate, the potential for these chemicals to move into crops and therefore to be ingested by people. And this is what we're going to look at here in Europe, and others have looked at this already in some detail. It's an active area of research at the moment. So those are what an antibiotic is, the range of chemistry broadly, how they get onto agricultural land, and what effect might these antibiotics be having on soil microbial communities. Well, certainly one can use methods in microbiology, culture-dependent or independent methods, to look at communities, diversity, identity, to try to link effects of these chemicals on organisms that have important ecosystem functions that we need to worry about. And so that's very much the ecotox approach that people can take. But in our particular instance, the endpoint of concern that we've been focusing on and that we continue to focus on is antibiotic resistance. And whether the entry of these chemicals into the environment is promoting antibiotic resistance, and therefore in human medicine, the risk of treatment failure and the consequences of human health. And I'll talk about that now for a moment. So again, antibiotics that first came on the market almost 100 years ago now many classes of antibiotics have come onto market from, say, the 1940s or so to more or less now. And if you look at the top left-hand corner of this image here, you'll see sulfonamides, and you'll see an arrow to the right of that word. The blunt end of that arrow is the year at which those antibiotics were introduced into the market. The pointy end of the arrow is the first year at which resistance was detected in pathogens. And so you can see for literally every class of antibiotics that's been brought to market, within a few years of introduction, you have resistance developing. And so the broad human health concern is that bacterial pathogens will accrue the genes that confer resistance to all these classes of antibiotics. And you have what's called pan-resistance. And if that's the case, if you get an infection with a pathogen that's pan-resistant, physicians no longer have a medicine available to treat you, and you are in big trouble. So it's currently estimated that there's roughly, this is 2019 data, there were roughly 1.2 million deaths globally that were directly attributed to a resistant bacterial infection, and roughly 5 million deaths that had some role for antibiotic-resistant infections and other comorbidities contributing to those mortality statistics. There was a review that came out in 2016 called the O'Neill Report, or review, that concluded using a modeling approach that by the year 2050, so what you see here is a list of concentric circles moving out from now out in time. And you see at the top right-hand purple slice of this pie, the estimate is by the year 2050, deaths due to resistant infections will be of the order of 10 million people globally, and that will outstrip all of the deaths due to all cancers, for example. So this is a huge problem, and people are very concerned about this and devising means of mitigating the pace at which antimicrobial resistance is developing. So within that broad context, our question has been, does exposure of soil microbiomes to antibiotics potentiate or make worse the environmental reservoir of antibiotic resistance genes or mobile genetic elements that increase the likelihood that a gene that's in a soil microorganism could potentially move into a human pathogen or an animal a pathogen, for example? And so that was our initial hypothesis. And is there any basis for hypothesizing that this might be the case? Why would one worry about this? Well, it turns out that the genes that are now in human pathogens were undoubtedly recruited from the environmental microbiome. That's because bacteria have been around for 3 to 4 billion years. And while they've been figuring out how to make antibiotics, they've been figuring out how to be resistant to those antibiotics. And here's a data set substantiating what I just said. So Jerry Wright's group from the University of McMaster in Hamilton, Ontario, took a core of permafrost from the Yukon, so in the northwest corner of North America. So this is soil that was deeply frozen for a long, long time, and took cores or slices of this permafrost core and identified DNA that was tens of thousands of years old on the basis of evidence like a DNA from animals that are long extinct being there. And then they sequenced that DNA and found an operon, so a series of genes that conferred resistance to the vancomycin class of antibiotics, and then synthesized that operon and put it in a bacterium and showed that the operon conferred resistance to vancomycin. So this is tens of thousands of years before organic chemists put vancomycin on the market or made vancomycin available on the market. And yet this DNA has been there for 30,000 years conferring resistance. So that's one example. Another example, and there are many, but this is work that we did a few years ago in Canada. And what we did was what's called a functional metagenomics experiment. So we extracted all of the DNA from soil samples, and we cloned large fragments of DNA in what are called phosmids, and we created a large library of soil DNA. And we put that library into an E. coli host that is sensitive to all antibiotics. And we plated that library, that E. coli clone library, on medium in the absence of an antibiotic. And of course, all of these different clones grew. And then we plated the library on a series of agar plates that contain various antibiotics at concentrations that would kill that E. coli isolate unless it had acquired a gene from the soil that conferred resistance. And through that experiment, we obtained a number of clone DNA fragments that conferred resistance to various classes of antibiotics. And what you see in the table here, if you look at the top left-hand corner, is a designation of a gene fragment. The soil from which it was obtained, that's not important, but the class of antibiotics that it confers resistance to. So this MB2MBL fragment had a gene or genes that conferred resistance to the penicillin type antibiotic. And then these other fragments, we detected resistance to sulfonamides, newer penicillins, aminoglycosides, tetracycline macrolides, pretty much every antibiotic that you'd be worried about. And then when we sequenced those gene fragments, the majority of them were identical to genes that were already in the public database that had been detected in pathogenic or commensal bacteria. So nothing particularly novel or interesting, just that these genes are already there in soil. But this a number of genes, including the one here that's highlighted with this red arrow, conferred resistance to, in this instance, the macrolide antibiotics. And the gene was not detected in any public databases. So we had no idea what this gene was or why it conferred resistance to the macrolide antibiotics. So Calvin Lau, the postdoc who did this work, subcloned this gene. So you see a series of horizontal lines here, and he gradually whittled away from the large fragment to a small fragment that still conferred resistance to the macrolide antibiotics. We sequenced that gene, expressed it, looked at the protein using a proteomics approach, and detected a protein that's very unusual and that has a large number of prolines in it. So prolines will put a 90-degree kink in a protein. So this thing basically looks like a corkscrew that you would use to remove a cork from a wine bottle. And it's totally unique. And we don't know what it does. But the point here is that there are genes in the environment that have yet to be detected in medicine, cryptic genes that confer resistance to really important antibiotics. So we need to think about what we're putting into the environment, because someday these things may be recruited through horizontal gene transfer, for example, and end up in human pathogens. So that's the take-home message on that one. So antibiotics in agricultural soil. So what would you need to think about if you're going to look at risk and how to manage that risk? And again, this is very similar to what you would do with a pesticide, for example. So the lower left-hand image here is you would look at how rapidly the molecule is destroyed. And if the molecule is very rapidly dissipated, you would conclude that your window of risk is very small. If the molecule is very persistent, you would be more concerned about risk. You would think about whether that molecule moves to crops that would be consumed directly by people or by animals that are then consumed by people. At the lower right-hand corner here, you would worry about, does it move to water that people would drink or bathe in or swim in, for example. And finally, you would be interested in whether that molecule is doing anything to the soil biology that you would need to be concerned about. And so let's talk about that now. And so the question is, what impact do antibiotics carry? So here, what I'm going to talk about is the use of fecal fertilizers. So what impact do antibiotics carried in animal and human waste have on soil microorganisms? And how would you look at that? What experimental approach would you look at that? So this is very complicated by the fact that sewage sludge or biosolids and manures are extremely complicated matrices. So unlike, say, if you're looking at the effect of a herbicide or an insecticide on soil microorganisms, where you have one chemical plus adjuvants maybe, here when you're applying let's say animal manure, you've got organic material, you've got inorganic nutrients, other things, metals. You've got enteric microbiology. If it's sewage sludge, you've got process microbiology. And so if you're interested in the impact of a particular chemical, it's very difficult or impossible to distinguish the direct impact of any one constituent on soil microbiology. And so recognizing this, one experimental approach that we've used are long-term antibiotic exposure studies that were done on our research farm in Canada. And so here the idea was to have a series of replicated small plots that are 2 square meters in area. And we applied chemistry directly to these plots, simulating what would be carried in animal and human waste, or potentially recycled wastewater. And so the advantages of using that exposure, that experimental approach, is we have very good control on the concentrations and the types of chemistry that we're adding. We have no interference from enteric microbiology. So if we see a gene increase in abundance in response to exposure to an antibiotic, we know that that gene was in the soil microbiology and not carried in E. coli or other things that we've added to the soil. We have no year-to-year variability in the input, say, as opposed to if you went to a sewage treatment plant every spring, it's not going to be the same in the second year as it is in the first year. The experiments are done outside real climate conditions. We can crop with something that we control, and the experiment can be very long-term. The downside of the approach is that the exposure scenarios are very unrealistic, obviously. We don't have enteric microbiology. So if your interest is, do we have horizontal gene transfer from E. coli that we've added to something in the soil? We can't look at that. We have only one climate and only one soil type. And so over the years, we started initially the experiment in 1999 with a mixture of chemicals that are used in pig production. So we're now in the 25th year of the experiment. Another whole series of plots were set up in 2010. So we're now, I think, in year 15 or thereabouts, with mixtures of different kinds of pharmaceuticals. So macrolide antibiotics, lincospecto, fluoroquinolones, cephalosporins, other things, coccidiostats, antiviral compounds, antifungal, anti-cancer, biocides, herbicides, other kinds of chemicals added to these plots. And so typically what we do is every spring, we take a soil sample, we apply the chemicals, we take a soil sample 7 days later, take a soil sample 30 days later, and these things are cropped to soybeans annually. We have 3 concentrations of exposure, so we have a control that doesn't see any drugs. We have 0.1 milligram per kilogram, which we consider to be an environmentally relevant concentration. We expect to see those kinds of concentrations with applications of human or animal waste. And we have an effect concentration, 10 milligram per kilogram or 10 ppm, which is higher than what we would expect to see with animal and human waste, but a concentration at which we might see effects. Just to emphasize that these experiments were undertaken in the Great Lakes Basin, which has a humid continental climate. If you look at the right at the image of the world, the purple areas are parts of the planet that have this similar climate. So parts of North America, going into Russia, into China, but certainly not in Greece, for example, or the southern parts of the planet. So you just need to think about that when you think about how broadly you would extrapolate the results. So the first question is, how persistent are these antibiotics? And I'll just give you some examples here. So in this particular experiment, we've added a mixture of 3 macrolide antibiotics. These antibiotics are very important. They act on the ribosome. And what you see here on the right is the concentration of the antibiotics in the field soil over the course of 5 years, the first 5 years of the experiment. And let's just look at the top left-hand panel. So this is erythromycin in plots that have received an initial nominal concentration of 10 milligrams per kilogram. And you see in the first year, you have a spike where we've added the chemicals and then it goes down. In the fourth year, it goes up, then it goes down. Fifth year, it goes up and goes down. So the take-home message is that these chemicals are not persistent in the soil. They will be degraded within the year of application. However, we found in the plots that have a history of exposure that we have evidence for enhanced degradation. And again, this is the macrolide experiment with soil from year 4. And what we do is we take the soil into the laboratory, we spike it with these antibiotics and watch how rapidly the antibiotics are removed. And let's look at the table. Let's just look at erythromycin. So the top row here, the control soil that is naive with respect to exposure to erythromycin. If you look to the right, the DT50 is of the order of 2 months or so. In those plots that have a history of 4 years of exposure, punctual exposure to 0.1 milligram, it's plummeted to 4 days, and 10 milligram 1 day. Similar results for the other macrolide antibiotics. Pretty good evidence that this is metabolism that's catalyzed by microorganisms. If we put into the soil erythromycin that's radiolabeled in one position at this methyl group that has the asterisks on it, and if you look at the top right, if you watch how rapidly that carbon atom is converted to carbon dioxide in the historically exposed soils, it's very rapid. In the control soil, it's much less rapid, and that's accompanied by rate of removal of total material as well. So good evidence for accelerated degradation of this antibiotic that would have the welcome effect of reducing the period of environmental exposure. We've seen this with other antibiotics as well. For example, the sulfonamide antibiotics, this other macrolide tyrosine. So we see evidence for biodegradation. In the case of the sulfonamides, we've isolated bacteria, characterized pathways, sequenced them, and showed that it's microbial biodegradation. However, some antibiotics are not amenable to enhanced degradation. Ciprofloxacin would be an example there. It's extremely sorptive to organic matter, so it's highly likely that it's not bioavailable in the soil, so no opportunity for an organism metabolizing it. So the second question we ask is, does exposure to antibiotics increase the abundance of antibiotic-resistant genes in a way that we need to worry about? And in the data that I'm going to show you here, what we did was we used shotgun metagenomics to evaluate the impact of antibiotics. And so what we did is we went back to this macrolide experiment. We isolated DNA following 10 years of punctual exposure from the control plots, the plots that had received the environmentally relevant concentrations, and the plots that had received the higher 10 milligram per kilogram concentration. And if you look at this cartoon in the middle of the slide here, you'll see a vertical line that has at the bottom the number 0, and this is the gene copy number in the control plots that had not been exposed to the antibiotics. The pink symbols are the plots that had received 10 milligram per kilogram, and the blue symbols are the plots that had received the environmentally relevant concentration. To the right of this, you see a number of designations for various antibiotic-resistant genes that we can detect by comparing the metagenome to publicly available databases. If you go back and you look at the pink symbols, look at the top of the image, you'll see that it is to the right of that vertical line. That is because that gene copy is more than 20-fold higher than it is in the control plots. And you can see there's a whole bunch of antibiotic-resistant genes that fall to the right of the control plot because they are more abundant. You can see that in very few instances with the blue symbols, the environmentally relevant concentration, do we see an increase in the abundance of antibiotic-resistant genes. So the primary conclusion from this kind of experiment would be at least in the agricultural context, an unreasonably high concentration will certainly increase the abundance of genes, including those that are clinically relevant, whereas the environmentally relevant concentration over a period of 10 years now does not do so. Another question would be, does exposure to antibiotics increase the abundance of mobile genetic elements? So these are bits of DNA that allow genes to move around within bacteria, within the genomes of bacteria, between bacteria of the same species, or potentially bacteria of widely divergent species. So if we see a large increase in mobile genetic elements in the environment, we would be concerned that this means that it increases the likelihood that genes ultimately can move to bacteria of human health concern. And this is data from the same experiment, the same bits of DNA. So what you see here, if you look at the middle column, are a number of different mobile genetic elements, integrons, transposase, insertion sequences, etc. And you could see with the pink symbols that the excessive concentration increases the abundance of many of these things, whereas the environmentally relevant does not seem to do so. So the conclusion is that the potential is there for these things being more abundant, but not at the concentrations that we would expect to see in the agricultural context. And finally, I think this is the last bit of data I wanted to show you, is we asked the question, once these soils have been exposed to these antibiotics for multiple years, if we stop adding antibiotics, how rapidly will these more abundant gene targets, MGEs, mobile genetic elements, antibiotic resistant genes, go back to background level? Is it going to be immediate, or is it going to take a long time? This kind of question is policy relevant, of course, because if one of your strategies for mitigating the pace at which antimicrobial resistance develops is to stop releasing antibiotics into the environment, you want to know whether or not that's going to be effective. And so in 2018 or 2019, actually, we stopped applying the antibiotics to these microplots. And what you see here on the top left-hand panel is the copy number of a gene called MSRE, which is important for macrolide resistance, expressed as copies per gram soil. And you see three horizontal lines. So you see the triangles are the copy number in those plots that have been exposed to 10 milligrams per kilo. And the two bottom lines are the control plots and the plots that have received only 0.1 milligram per kilo, which I've already told you has not increased the abundance of gene targets that we've looked at. But you can see here that 4 years after we stopped adding the chemicals, the copy number of this gene is still much, much higher in the historically treated plots than the control plots. Same thing for this other gene, MPHE, also important in macrolide resistance, and in integron 1, which is an important mobile genetic element that's very responsive to anthropogenic impacts. So the conclusion from this is that 4 years after we turned off the taps, and even though I told you earlier that these chemicals are biodegradable, we don't see these gene targets going back to baseline. And we don't know why yet. We don't understand why. But I would argue that this is somewhat concerning. And I'll just leave it at that. So that's the last piece of data that I wanted to show you. And I wanted to finish just by plugging this conference, which is called the Environmental Dimension of Antimicrobial Resistance Conference, which will be held in Montreal in Canada in May of 2024. And if you have an interest in any environmental aspect of AMR, whether it's on the chemical side or the microbiological side, I would encourage you to come. And if you take a photo of that code there, it'll take you to the website. And with that, Dimitros, I'm finished, ready to answer questions if there are any.
Excellent. Thank you very much, Ed, about this really inspirational presentation. Very, very nice insight into the interaction of microbes with antibiotics in soil. Are there any questions from the audience? Because I have some questions. Always have questions for Red. So please switch on your microphone and
I'll stop sharing my screen.
I can start maybe and trigger a bit of a discussion. Right. Okay. Oh, Sotiris, yes, Soto.
Thank you very much for this very inspirational and amazing talk. I was just waiting whether any of the other participants were going to ask. I'm curious, let's say, what happens with the microbial loads of the inputs in the agricultural soils, according to based on your experience? And what, if you can say something about, let's say, microorganisms that may act as reservoirs of microbial resistance, let's say, to antibiotics?
So for sure that the microbial world, whether it's soil or aquatic systems, has these antibiotic-resistant genes. And so the concern is whether waste streams that we're putting in the environment, whether it's human waste, animal waste, whether it's effluent from pharmaceutical manufacturing companies, is increasing the abundance of these reservoirs of mobile genetic elements or antibiotic-resistant genes. And it certainly appears to be the case that this is happening. So the conclusion from these kinds of data would be that we need to reduce entry of these chemicals into the environment. And if this is something that you're interested in, I was involved in a recent report from the United Nations Environmental Program that goes into this in more detail. If you Google UNEP and superbugs, it'll take you to that report that describes the issue and has a number of recommendations on how we can deal with this. The other interesting part of this science, to my mind, is the concern that we're actually increasing the rate at which microorganisms are evolving through the entry of these chemicals into the environment. So through lab-based type studies, we know that bacteria that are exposed to chemicals that create reactive oxygen species will accelerate horizontal gene transfer. So the rate of plasmid movement or transduction, so viral mediated DNA movement, can increase in populations that where you have chemicals that are causing reactive oxygen species. Likewise, the fluoroquinolone antibiotics that interfere with DNA synthesis will set off what's called the SOS response. And what that means at the end of the day is that the fidelity of replicated DNA decreases because the organisms are trying to go through more mutations to find one that will allow them to survive under those circumstances. So you have accelerated rate of DNA exchange and accelerated rate of mutagenesis, therefore increasing the rate at which microorganisms could develop or exchange genes that are involved with antibiotic resistance. So I'm not sure if that answers your question at all, but I've tried.
It was great answer. Thank you very much.
Right. Anyone else wants to ask question to Ed? Please raise your hand and switch on your microphone. Right. Okay. I have a question maybe. So I see that what my main question I would say is that you see a lot of antibiotics are prone to enhanced biodegradation in soil, and you've been involved in isolation of bacteria that they degrade sulfonamides, for example, and other antibiotics as well. So the question is, which comes first? So which phenotype comes first? I would expect evolutionary that first the bacterium should have some sort of a resistant mechanism towards the antibiotic, and then it has the luxury to start consuming it. Is that what you see, or what you expect?
So it would depend on the concentration of the antibiotic. So let's say you've got a sulfonamide degrading microorganism that has a gene that confers resistance to sulfonamides, and it's got a catabolic pathway that will allow it to grow on that chemical. If the sulfonamide concentration is very high to the point where it's toxic, maybe that antibiotic resistance gene is required. If the sulfonamide concentration is below that toxic threshold, but still provides enough carbon and energy that it's interesting, you would still use the catabolic pathway to grow on. So I think it depends on the concentration of the molecule. I will say that a number of people have talked about catabolism as potentially being a means of antibiotic resistance that's of medical concern. There are no instances where metabolism of any antibiotic is the mechanism for resistance in the clinical setting. I'm guessing that that won't happen because you need multiple genes. What you want is a mechanism that involves one gene, an efflux pump, or some way to modify the antibiotic, or some way to get around it that's much easier, simpler. It doesn't require more complicated things than otherwise. What I find quite interesting in this is, why would an organism in nature bother evolving a catabolic pathway for an antibiotic that's used in human medicine? Why would a bacterium that lived 30,000 years ago bother making an operon that confers resistance to an antibiotic long before people are putting these antibiotics in the environment? Well, that would suggest to me that the antibiotic concentrations in the environment are sufficiently high, in some environments, sufficiently high, that it warrants having an antibiotic resistant gene, or it warrants having a catabolic pathway, because that confers you an advantage in an environment that's otherwise carbon limited. So if you think about the scale of a biofilm, which is the scale at which microorganisms live in the real world, whether it's a soil particle or a sediment, at that scale, could you have antibiotics at concentrations that make it worthwhile metabolizing them, or make it worthwhile necessarily having an antibiotic resistant gene? There aren't any of that data available to my knowledge. Nobody, to my knowledge, has quantified antibiotics at the micron scale. But we recently did a modeling exercise where we looked at the antibiotic fermentation literature, so the literature from the industry that produces antibiotics. And we looked at the rates of antibiotic production in fermentations. And through that, we calculated, in many instances, how rapidly a bacterium produces an antibiotic. So many grams per hour per cell. Turns out it's about 10 to the minus 15 or so grams per cell per hour. And then we did a modeling exercise using Fick's law. If you assume that the antibiotic is produced at that rate, what would the concentration be within a micron of that bacterium, within 10 microns, between 100 microns? And what might those concentrations be? And we made a whole series of assumptions that may be totally wrong, but the bottom line is that aggregates of a few hundred cells, if we're correct, are producing antibiotics that are in the microgram or even milligram per liter range, the range at which you would want to be resistant, or it would be a very useful carbon source. So I find that quite interesting. I find that as environmental microbiologists, often we think about a kilogram of soil, a liter of water. We're less confident thinking about the scale at which microorganisms really live, whether they're exposed to a pesticide, or producing an antibiotic, or consuming an antibiotic, or needing to be resistant to the antibiotic.
Yeah, I think this is a matter of technicality, really, because it's not very easy to measure at real micro scale what is the environment, the microenvironment that microbes are exposed to.
Yeah.
So I have a second question, but I will wait if someone else is coming up with a question. So I'll ask that as well. So you've isolated, and there are a lot of examples now in the literature that there are a lot of available isolates actively degrading veterinary antibiotics. So I was thinking that for veterinary antibiotics, one way to stop, to break the release, the environmental dispersal their dispersal in agricultural soils, is to actually maybe intervene at the stage of the manure, which is a major, I would say, source for their release in the environment. So I was thinking that one way to do it is to bioaugment these manures, maybe, with antibiotic degrading bacteria. Because in our hands, with what data we have, but also the literature, it seems that other type of treatments that manures are going through, like anaerobic digestion or composting or stockpiling, they don't seem to affect at all the dissipation of antibiotics. They seem to persist there. So I was thinking, what do you think about bioaugmentation, using these tailor-made inocula for bioaugmentation of manures, and that way we break actually the root, stop the root?
Yeah, so I would say, what would the business case for that be? Why would a farmer buy bacteria and stick it in the manure lagoon, for example? There would have to be a business case that would have to be, I guess, driven by regulation. So of course, this is an idea that people have had, probably a good idea, might be effective, but I just don't see the business case for it necessarily. What I would argue in the animal context is the best strategy is to devise means of keeping the animals healthy so that the need for antibiotics goes down. And the advantage of that is that you would have fewer release of residues into the environment, and you would have less exposure through the food chain. So you're intercepting two potential transmission pathways back to people. And so the strategy there would be, specific for chickens or pigs or whatever it is, vaccination, nutrition, everything that you need to do so that the need for antibiotic use is reduced. Ideally.
All right, so you think that the intervention at the source, really at the use, might be the best way to actually reduce, at least reduce the release.
Yeah.
Right. Yeah.
And that also has marketing advantages in some instances.
Certainly.
So it's probably the case in Greece and elsewhere that people want to eat antibiotic-free chicken or whatever it is. So there's a marketing profitability advantage to the producer if they can do that as well.
But one thing that I might I would like to bring up also is the environmental risk assessment of veterinary drugs, because I'm pretty sure you know that the level of environmental risk analysis that is done, the depth of environmental risk analysis that is requested for veterinary drugs in general is quite a light one compared to what we have for pesticides. So we, at tier 1, you have to answer 20 questions. Most of the questions, most of the risk assessment stops in the first 5 or 6 questions, and then everything ends up in the market. So, and I found personally that when you go and talk to veterinary medicine people about environmental risk assessment of veterinary drugs, they are not really very keen on listening about it, and wondering if you have had this experience, because you are working on this as well.
So what I would say is, to my knowledge, in no jurisdiction is antimicrobial resistance, as I've described it today, a metric that is involved in the environmental risk assessment. Nobody has asked, okay, if this chemical goes into the environment, am I going to increase the reservoir of antimicrobial resistance, and therefore maybe I have a public health issue? So that's just not done. The second comment I would make on this topic is that in these long-term field experiments that I've described, and I haven't gone into them in detail, we would observe an increase in the gene targets that we can measure by qPCR maybe 2-3 years into the experiment, not within the 30-day window in the first year. So there's a rate of microbial evolution and changes in community composition that results in a change in the phenotype, in this instance, antibiotic-resistant genes, that's only detected over the space of many months or years, at least in our climate conditions. So that means that the very quick, OECD or whatever level test it would be, which is I put my chemical in a soil sample, and a month later I wait to see if I have all kinds of antibiotic-resistant bacteria, just would not capture that, because we're not working at the pace of microbial evolution. It's the same thing with enhanced degradation of pesticides. You might not see it in the first year, so you might say, I'm not going to have any enhanced degradation, but you might see it in the second or third season.
Yeah, that very much depends on the type of chemical also, I suppose.
Yeah. So it's a problem.
Yeah. All right. Okay. Any more questions for Ed? Because it seems like I have monopolized the discussion.
I have one.
Hi,
You kind
Alex.
of mentioned about the climate condition.
Yeah.
And you show most about the data regarding the north hemisphere.
Yeah.
What do you expect in the south, like with hot conditions, and kind of the regulation is there's no existing regulation, and farmers, they use whatever they want, and the amount also is the same thing.
So with respect to south versus north, which I think means, let's say, tropical conditions or versus humid continental climate, I mean, you would expect chemicals to be broken down more rapidly, I guess, because temperature is typically the key rate controlling parameter. Assuming that other environmental conditions such as moisture content are not rate limiting. So I would just make that general comment. With respect to the amount of, I guess, manure that fertilizers that farmers are using, you have to do think about a risk-benefit analysis. So you have all the benefits of recycling these organic materials onto land with respect to NPK and organic carbon and water holding capacity, versus what I'm showing here, which is at the concentrations that we expect these chemicals to be in a plowed field that has received manure or biosolids, we don't detect an effect. You need to do a risk-benefit analysis. I think we need much more of this kind of data in different climate conditions, personally. But these are farm, with some kind of secure funding, et cetera, et cetera.
Mm-hmm.
Anybody else? Yeah, I think that these long-term experiments are really very, very, very important. You show very, very much the benefits that you get from these long-term experimental fields that you have, and you can get access to these fields, and you can do all sorts of different things. But these are extremely important to have in different actually continents and different under different climatic conditions, and see how this would behave, I would say.
Yeah, so I haven't thought about this at all, but if Horizon Europe or some other kind of global initiative could be used as a framework to have people doing these kinds of experiments in different places, and then having common metadata, common analytical approaches, et cetera, maybe that would be a way to extrapolate whether what I'm saying, what we've observed in Canada, is relevant for Africa or Southeast Asia or South America or Greece or wherever it is.
And the applications you've been doing, Ed, on these plots is direct application of the antibiotic without actually the manure substrate.
No.
All right, okay. Because I suppose that will have changed a bit the implications and the response of the soil microbiota as well.
Yeah, it makes it incredibly complicated for a number of reasons. And one of the things that we're thinking of doing in the project that we're starting here is modeling the bioavailability of antibiotics once they go into agricultural ground. Similar things to what people do with pesticides, with pedotransfer functions and those kinds of things. But if you think about adding a chemical directly versus in sewage sludge, that changes so many things, and I don't know how to how to incorporate that into a model.
Yeah,
Like
that's quite complicated. Because I suppose you add a lot of fresh organic matter there with your antibiotic in the soil, and a lot of microbes that they are mostly enteric microbes that they are not really happy in the soil sometimes.
Yeah. Complicated. So you always have this tension between having a research question and needing to take an experimental approach that is reductive enough that you can get an answer to your question, but then it's, the criticism is it's so unrealistic that it doesn't represent what happens in the real world.
Yeah, I think you have to set your priorities really. Right. Any more questions for Ed? No. So maybe I will, if there are no more questions. Thank you very much, Ed, for the really nice presentation.
Thanks again.
It was a real pleasure. And since you are in Dijon now and you will be around